In this video, we will explore how the genetic code within DNA translates into proteins that perform important functions in our bodies. Let's begin by outlining our key objectives. By the end of this video, the goal is that you will be able to describe the structures and functions of DNA and RNA, understand the process of transcribing DNA sequences into RNA. Finally, use an amino acid chart to translate an RNA sequence to amino acids. Objective 1. The basics of DNA and RNA. DNA, or deoxyribonucleic acid, is the blueprint of life, containing the instructions needed for an organism to develop, survive, and reproduce. RNA plays a crucial role as the messenger, carrying DNA's instructions for protein synthesis. The process of using DNA to make proteins is called protein synthesis. To help an organism survive, DNA sequences must be converted into RNA. RNA must then assemble amino acids to produce proteins. Proteins are like magical workers inside of our body. They play a key role in digestion with enzymes by breaking down food for nutrients. Proteins also act as superheroes that protects our bodies from germs. They defend us with antibodies in the immune system. Additionally, they help our bodies send and receive vital messages as hormones to regulate bodily functions that control our health and mood. DNA is made of 4 chemical bases, or nucleotides: adenine, guanine, cytosine, and thymine. Like a puzzle, due to their molecular shape, adenine pairs with thymine, and guanine pairs with cytosine. We call this complementary base pairs. DNA strands are organized in sets of 3 bases called codons. Each group of 3 base pairs is a single codon. RNA is also made of 4 base pairs: adenine, guanine, cytosine. Although instead of thymine, RNA uses uracil. Because of their similar shape, adenine pairs with uracil, guanine still pairs with cytosine, and their bases in RNA are still organized by codons. Objective 2. Accurately transcribe DNA sequences into RNA. The first step in protein synthesis is called transcription. It occurs in the nucleus of a cell. During this process, DNA is unzipped, and the mRNA strands copies a strand of DNA using complementary base pairing. A pairs with U, T pairs with A, C pairs with G, and G pairs with C. Let's translate the first codon, CGT, from DNA into RNA using the base pair rules. The first letter, C, will pair with G. The second base, G, will pair with C. Finally, the third letter, T, will pair with A. Moving on to our second codon, GTT. G matches with C, T with A, again T with A. So GTT becomes CAA in our RNA language. For our last codon, ATA. A matches with U, T matches with A, and A matches with U. ATA became UAU. Now you will practice decoding DNA into RNA using this DNA sequence. Your job is to match the DNA bases to their RNA partners. Remember to take your time and use the chart. When you are done, check your RNA sequence to make sure it matches the rules of base pairing. Objective 3. Utilize the amino acid chart to translate RNA sequences into amino acids. The second step of protein synthesis is called translation. During this step, mRNA leaves the nucleus and goes into the cytoplasm. mRNA will then attach itself into a ribosome. The strand of mRNA are read 3 bases at a time. tRNA, or transfer RNA, brings the amino acids to the ribosome to help make the protein. Here is our universal genetic code chart. It's like a decoder for our mRNA message. Each 3-letter mRNA codon matches with 1 amino acid. First, we will transcribe the DNA strand to RNA. TAC now becomes AUG. In the chart, we will look for the first letter in the mRNA codon in the left column, labeled first base. For the codon AUG, that first base is A. Now, we're going to look for the second letter at the top row. Our second letter is U. Finally, we will match our third letter with the boxes that intersect our first 2 letters. The third letter for AUG is G. There we have it. AUG codes for the amino acid MET, which is also the amino acid that signals the start of protein synthesis. Let's do another example. TTA transcribes to A, and its amino acid, using the chart, is LYS, LYS. Final example. CCC transcribes to GGG. The amino acid for GGG is GLY, GLY. Congratulations! You are now decoding the language of life, translating RNA into proteins. Keep practicing and you'll be a master of the genetic code. In this guided practice, your job is to transcribe the DNA bases into RNA and then locate the amino acid for each codon using this chart. Big takeaway. Before this video ends, it's important for you to remember that DNA makes RNA. RNA makes amino acids, and amino acids make proteins. Great job making it to the end of the video. Please use this checklist to reflect on your understanding of the learning objectives.